Good afternoon, everyone. I'd like to start by thanking Laura and Thomas for organizing this very interesting workshop today. I'm not going to talk so much about connectivity per se, but more about how do we think about the neural mechanisms and neural network mechanisms giving rise to brain electrical field potentials that we record with MEG and EEG. And hopefully, as you will see, this will have implications about how we interpret results obtained with connectivity analysis. These days, a student who comes into neuroscience often learns that in single and multi-unit neurophysiology, neuronal activity, neuronal spiking activity is person-like and irregular. On the other hand, brain electrical field potentials recorded by local field potential, EEG and MEG signals, contain mostly brain oscillations, which are periodic signals. So why is there this discrepancy? Let's look a little bit deeper. In unit physiology, we know that most pyramidal neurons fire in a person-like manner, or at least irregular manner, such as this black, do you see my mouse here? Oh, here, okay. So such as this black neuron here recorded from the primary motor cortex in the macaque. There are also certain, sorry, if we go, can we go back? Oh, how do I go back? Oh, yeah, okay, sorry. Okay, so there are also a minority of pyramidal neurons that fire in a clock-like manner, such as this red neuron recorded in the same region. However, the majority of pyramidal neurons, which are the neurons generating an open electrical field, which we can see with EEG and MEG, as well as ECOG, fire in an irregular person-like manner. So how do we resolve this discrepancy? So one possibility is that because of the network mechanisms, such as the interactions between excitatory and inhibitory neurons, the EEG and MEG signals really see the network phenomenon, which is mostly made up of oscillatory activity. This is to some extent the case because we know that the interaction between inhibitory neurons and inhibitory excitatory neurons can give rise to brain oscillations. But as I will show you, this is not all of the story. So in fact, arrhythmic activity is prevalent in brain electrical field potentials as well, including local field potential, ECOG, EEG, and MEG. It's just that we have known and studied about them much less than brain oscillations. So in the past, it used to be ignored, and now more and more studies have been focusing on them. But we still have a lot to discover about what's the mechanism generating them and what are their functional significance and functional mechanisms. For example, these are, is there a pointer? Oh, okay, sorry. So for example, these are 3 electrodes, ECOG electrodes recorded from one patient. This red electrode, as you can see, has delta oscillations and beta oscillations. And this blue electrode has theta oscillations and beta oscillations. And the black electrode has just these irregular fluctuations. If you look at their activity in the power spectrum in the frequency domain, you'll see that all of them follow this 1/f-like pattern, which means that power falls off as inverse of frequency to an exponent, which shows up as a roughly linear plot on a log-log plot. And on top of this so-called 1/f spectrum, there are peaks at different frequency that correspond to different brain oscillations. So here are the take-home messages I'd like to have you walk away from this talk with. So first of all, arrhythmic activity is prevalent in brain electrical field potentials. And this activity is likely contributed by asynchronous neuronal firing in recurrent balanced cortical neuronal networks. And lastly, arrhythmic brain activity influences behavior performance and contributes to the processing of natural stimuli. So again, let's look at the phenomenon a bit closer. So these are about 10 seconds of ECOG recording from 100 also electrodes. You can see that there are some oscillatory activity in the delta range, the mu rhythm here in some electrodes that rides on top of these constant irregular fluctuations across all electrodes. And again, in the power spectrum, we see, so across all of the electrodes, we have this 1/f pattern with oscillatory peaks riding on top of it. So in fact, this kind of power law distribution of power spectrum is very prevalent in the brain across multiple spatial scales. So with local field potential, it's been reported by multiple labs that you see the same 1/f-like activity pattern. This particular recording doesn't have much oscillations, but oscillations come and go, and 1/f activity is common across most recordings. In the ECOG, and by the way, the exponent, power law exponent, which describes this slope in the log-log plot, is about -2 for local field potential. This is a study from Christoph Koch's group. And with ECOG, the exponent, the slope in the power spectrum is around -2.4. So in two studies, in my own lab and in Carl Miller's study, both studies reported very similar power law exponent around -2.44, -2.46. So it's a very robust and reproducible finding. So in the in vivo recordings of neuronal membrane potentials from Alan Dextek's group, we also know that neuronal membrane potential fluctuations have a similar 1/f-like power spectrum with a similar exponent. This is a finding reported from the leech nervous system from the neuronal ganglia using the intact animal and the intact ganglia as well as the isolated ganglion. So what they found was that when the network becomes segregated, so that when the network becomes when they dissociated the subcomponents of the network, this exponent became shallower. But overall, the network spiking rate, which is the population neuronal firing rate in this network, has a power law distribution. As I'll show you later in a computational study, I think we now have a plausible mechanism linking the population firing rate 1/f activity in the population firing rate to EEG and MEG signals, and a plausible mechanism explaining the difference when the network topology changes, the difference that generates for the activity power spectrum. So lastly, Klaus Lincoln-Hansen and colleagues and other people have a long established line of research showing that if you look at the power amplitude fluctuation of these brain oscillations, they too have a 1/f type of power spectrum, as shown in the open circles here. And the filled dots on the bottom here is from an empty room recording, showing that the instrument noise, which is sometimes a worry, really doesn't contribute much to the recording. So the phenomenon is very robust, very reproducible. How come we as a field have thought about it much less than the oscillations? One problem that has deterred people from taking this activity seriously and really digging into it, at least some years ago, was the worry that it's really, that it may be homogeneous, non-specific 1/f noise that is prevalent throughout nature. For example, physicists have described that many dynamical stimuli in natural environment, such as seismic waves, wind velocity, etc., have a 1/f type of power spectrum. Stock market has a similar power spectrum. This 1/f squared power spectrum is also called random walk. So many different systems have a similar 1/f-like power spectrum. So could it be that this is an unspecific universal noise? So when I was a student and first got interested in this phenomenon, I discovered something quite interesting. So when something like this 1/f spectrum is present across a variety of systems. Physicists tend to get excited about it, thinking that there may be a universal mechanism behind this phenomenon. Whereas on the other hand, biologists tend to get scared away from it, thinking that this is non-specific, can't be important. So the truth is probably somewhere in between. So we have to remember that the power spectrum does not tell us all about the property of a signal. It's only a second-order statistical structure. Beneath this second-order statistical structure, there may be higher-order statistics, finer-grained spatial temporal patterns that are unique to each signal. So I'll come back to this in a couple slides. So another worry that has deterred people from studying this arrhythmic or broadband activity is a worry that maybe a lot of it is contributed by instrument noise, because 1/f type of dynamics are also present in, say, electronics. So as I showed earlier from Klaus Lincoln-Hansen study, the empty room recording shows that the instrument noise is orders of magnitude lower than the activity we record from the brain. This is the same case for the ECOG power spectrum I showed you earlier, where the brown trace is a dummy recording looking at the instrument noise. This is log-log plot, so this is really orders of magnitude difference in the amplitude. So coming back to the idea I mentioned a second ago about there being higher order statistical structures unique to each signal that might be hiding beneath the universal 1/f power spectrum, we looked into this some years ago using a method borrowed from oscillations called nested frequencies, and many people here have also done illustrious work with this method. So the idea is that we filter this broadband activity to different frequency ranges, and we extract the phase from each lower frequency range and the amplitude from each higher frequency range, and look at how they co-modulate. In other words, whether the phase of a lower frequency might co-modulate with an amplitude of higher frequency. And we found very prevalent structure, especially during the awake state, less so in slow wave sleep, but there's still some structure there. By contrast, classic Brownian motion, or random walk, has no nested frequency pattern. So on the top, this shows that with a power spectrum that has both 1/f broadband activity and brain oscillations, there is a method for decomposing them, at least in the domain of power spectrum, using a method called coarse-grained spectral analysis. So the intuition is that if you have an oscillation in the spectral domain, if you rescale it, like sample every other sample or sample twice as fast, you will move it to a different frequency. And the cross spectrum of this rescaled signal and the original signal will be flat, will be 0. Whereas 1/f signal, because it's scale-free, after you rescale it, the cross spectrum is just a scaled version of the original one. So you can separate this, the original spectrum into the broadband arrhythmic component and brain oscillatory component. And when we do that, we see a tripartite shape in the broadband component of the ECOG signal. And I'll come back to this later, where we now have a computational mechanism that can explain this particular shape. So, okay, so let's carry on with this nested frequency observation. When we do the same analysis with stock market and Earth seismic waves that share an almost identical power spectrum as the ECOG signal, we also find very prevalent nested frequency patterns. So in these signals, if you look a bit deeper at how the high frequency amplitude modulate at different phases of the low frequency, you'll see different patterns between stock market and Earth seismic waves. So for example, in seismic waves, high frequency amplitude tends to be higher when the low frequency phase is at plus minus half pi, whereas stock market shows a different pattern. And when we look at ECOG signal recorded from human subjects, we see yet a different pattern. So the presentation here is different because these two are just a single time series, whereas for the ECOG we have several hundred electrodes. So each dot here is one electrode, and each dot has a phase modulation graph like this. And we've taken the amplitude, the phase of the low frequency, where the amplitude of high frequency is the strongest, and plotted that on the x-axis. And what this pattern shows is that across hundreds of electrodes, high frequency amplitude tends to be stronger when the low frequency phase is at plus minus pi. That is at the trough of the low frequency. So initially, when we had this observation, we simply concluded that beneath the common power spectrum, each signal has its unique fingerprint that can be seen as different nested frequency structures. Several years later, we realized what could be the mechanism behind these individual nested frequency patterns. For example, in ECOG signal and local field potential as well, oftentimes you have these sharp transients in the signal. And EEG and MEG signals, in EEG and MEG signals, this has also been shown, although in MEG signal, this is more symmetrical, both positive and negative. And what's driving this is the population firing bursts in the underlying neuronal network. So when neurons in the underlying network fire synchronously, that drives the current going into the neurons, going into the intracellular space, and drives sharp transients in the electrical field potentials. And these kind of negative transients would give you a nested frequency pattern like this, such that high frequency amplitude is higher at the phase plus minus pi of the low frequency. On the other hand, Earth seismic waves often have step functions because of the tectonic plates bumping into each other, and this gives you this sort of nested frequency patterns because this transient is related with phase plus minus half pi. So this is fancy statistics telling us something that we can actually see with the naked eye. So when we look at these different signals, they all share the same 1/f² type of power spectrum. In the time domain, we can see that the signals have different waveforms that we can pick up with the naked eye. So, so ultimately, this says that there are this 1/f activity in the different systems have different higher order statistical structures, and there is not necessarily a universal generative mechanism as physicists thought many years ago. So next, I'd like to talk about functions of scale-free brain activity. So coming back to the ECOG power spectrum, it is now well established that the high-frequency band of this so-called broadband gamma range, the power of that signal correlates with neuronal spiking rate. And the low-frequency range, so-called slow cortical potential, is known as the shift, negative versus positive shift, and the phase of this signal carries a lot of functional significance. So let me first tell you a little bit about what we know about the physiology of the high-frequency broadband power. So my Kahane's group had a very nice study showing that the high-frequency broadband power correlates with the population neuronal firing rate in human subjects. This is a very nice monkey study from John Manso's group showing that the broadband power and oscillations can be dissociated. For example, in V1, when you increase the visual stimulus size, you have increased gamma oscillation power, but decreased broadband power. And at the same time, you have decreased population firing rate. So again, population firing rate and broadband power go together and they can be dissociated from the narrowband oscillations. And in this high-frequency range, you can also, in some cases, find changes in the slope of the power spectrum, which is an index of the strength of temporal correlation in the signal. And then in the low-frequency range, several years ago, we found that the low-frequency power as well as the slope in this part of the power spectrum can be modulated by task performance. So when you perform a task, the power law exponent in this range decreases, and a reduction of power law exponent is indicative of reduction of autocorrelation, suggesting that the signal has less temporal redundancy over time. This is a phenomenon that I'll come back to later in a computational model. So let me summarize this first part. So hopefully I've shown you that scale-free dynamics is prevalent in the brain across multiple levels. It contains rich temporal structures manifesting as nasty frequencies. And beneath an identical power spectrum, the fine-grained temporal structures could vary from system to system and signal to signal. And this signal, both its power and its autocorrelation statistics, can be modulated by task performance. So what is the source of broadband arrhythmic electrical field potentials? Several years ago, together with Xiaojun Wang's group, we constructed a neural network model to try to shed light on the generative mechanism of this signal. So we used a very standard recurrent neural network model with minimal and plausible, biologically plausible mechanism. So here X is a vector that describes a set of nodes that can be neurons or neuronal groups, and they are recurrently coupled with a random connectivity matrix, W. And each node has a leak term, which is the time scale of the autocorrelation of the activity in each node. And we found that, so this is a very minimal recurrent random connectivity matrix. So we found that when this network is balanced, in the sense that excitatory activity balances out the leak plus inhibitory activity, it produces a power spectrum that is very similar to the human ECOG power spectrum with this tripartite shape. So now with the model, we can tease apart what are the mechanisms. So this Ni around 1 hertz, 1 to 10 hertz, is contributed by the NMDA receptor's time constant in the network, which is relatively slow, but biophysically plausible. And what's really been a mystery until we constructed this network was what is contributing to this low frequency, slow cortical potential. And we found that recurrent activity in the network really contributed to this part of the signal. So what's nice was that the network could recapitulate could capture the empirical finding we found of the changes in power spectrum between states. As I mentioned earlier, during tasks, we found reduction of power as well as autocorrelation in the lowest frequencies. And this was captured in the network when we reduced correlated input into different nodes. So in other words, this recurrent network converts spatial correlation into temporal correlation. And when different nodes become more independent, as often happens during task activation, the low frequency power and autocorrelation is reduced. It also captures changes between states, arousal states. So in the empirical data, we found that during slow wave sleep, the low frequency power is higher and there is lower autocorrelation in the signal. And in the network, we found that when we reduced effective connectivity across long distance, and this is a finding we know that happens in slow wave sleep from the very nice work from Giulio Tononi's group, with that mechanism can recapitulate the same change in the empirical power spectrum. So one thing that I'm really excited about the insight we gained from this computational model is a mechanism that can potentially explain differences between EEG, MEG, and ECOG power spectrum. So this is just example from my own lab. These are single subject power spectrum in resting state across all of the electrodes or sensors we recorded. So as you can see, oscillations are much more prominent in EEG and MEG signals than ECOG. And this is sort of implicitly known in the field because 1/f activity, broadband activity, really got taken notice of when people started doing serious ECOG studies. So Mike Kahana, myself, Camilla, who worked on this, have all worked with ECOG to begin with. So what's causing this difference? So in the network, what we found was that when we calculate the population firing rate by different degrees of spatial averaging, this high frequency broadband power reduced, the more spatial averaging you did. What this means is that what this points to is a potential mechanism explaining this difference. So in EEG and MEG, it's possible that this spatial averaging reduced the broadband power in the high frequency range, rendering the brain oscillations riding on top of it more prominent. So to summarize this part, we found that a balanced recurrent network reproduces the power spectral shape of human ECOG. This network converts spatial correlation to temporal correlation, providing a potential mechanism for the reduction of power law exponent or autocorrelation. The two are basically equivalent during task. And changes in network functional connectivity structure could account for changes in the ECOG power spectrum during slow wave sleep. And lastly, spatial averaging could potentially explain why brain oscillations are more prominent in MEG and EEG than in ECOG. So in the remaining time, I'd like to tell you about some more recent work about the potential functional role of arrhythmic brain activity in processing natural stimuli. So as I mentioned at the beginning of the talk, many natural environmental stimuli has prevalent 1/f dynamics, showing up as a power spectrum in the temporal, showing up as a 1/f type power spectrum in the temporal frequency domain. So for example, this is a wind velocity, and on the bottom, this is loudness and pitch fluctuation of urban soundscape, so urban auditory environment. Same thing happens with if you record in the rural environment. I've mentioned that brain activity has prevalent 1/f activity, and it's been known for a couple decades that human behavior fluctuation often exhibits 1/f type of fluctuation as well. And it's very well established that speech and music in their loudness fluctuation and pitch fluctuation follows 1/f spectrum. So it's possible that all of these are related somehow. So in fact, there has been a very nice series of work, especially from Matthias Pava and Klaus Lincoln-Heyer-Hansen's group, that show that 1/f activity in the brain can influence and even underlie the 1/f behavioral fluctuation in human subjects' behavior performance. So we did a couple studies on this as well, and I'll talk more about some of these findings at an afternoon symposium on Wednesday. And I'd like to tell you about some very recent work from our group about the 1/f brain activity's potential role in processing and predicting natural environmental stimuli. So the motivation for this is that because the statistical structure of 1/f activity is similar to these natural environmental stimuli, maybe it is especially suited for processing these stimuli. So to look at this, we constructed auditory tone sequences that follow 1/f type of fluctuation. Each trace is a single tone sequence, and the y-axis is the pitch fluctuation over time. And these vary from white noise all the way to Brownian motion. So the power law exponent ranges from 0 to 2. So I'll just play one of these, some of these for you. Can we turn the volume up? Could we turn the volume up? Okay. So this is a white noise-like stimulus. And this one is more like a Brownian motion. It's much more melodic. So this is purely computer-generated for the aficionados, fractional Gaussian noise and fractional Brownian motion. And so we recorded MEG activity while human subjects listened to the stimuli and looked at which component of MEG signal really processes this natural stimuli. So first of all, human subjects can discriminate the sequence autocorrelation structure. So these are the stimulus autocorrelation, power law exponent from 0 to 2, and these are the response. So human subjects can discriminate this pretty well. And so we asked which kind of neural activity tracks these sort of naturalistically evolving stimulus sequences. So this can be rephrased as the following question, which is directly analytically tractable. Which kind of brain activity is reliably modulated by repeated presentations of an identical tone sequence? So, for example, when we present the same sequence many times versus sequences that are different but statistically similar, which kind of brain activity is reliably modulated across trials in this case, but not this case? So statistically similar means similar autocorrelation structures and similar variance. So to look at that, we can use across-trial phase coherence. We've heard about across-regional phase coherence from some of the earlier talks today. So across-trial phase coherence tells us at each frequency how reliable is the phase of that frequency across multiple trials. And we can do this for both the identical set of trials and for similar set of trials. And then we can calculate the difference between them called phase dissimilarity. So this tells us the reliable modulation of a particular frequency band for identical repetitions of the same sequence above and beyond statistically similar sequences. So phase dissimilarity in the low frequency range has a tuple like this. This is significant, in fact, across most of the sensors across the scalp, and has a bilateral distribution of auditory cortex. So when we look into the phase spectrum, phase dissimilarity spectrum, we find that it's mostly contributed by arrhythmic broadband activity in the low frequency, and it has a dip at the frequency that corresponds to tone presentation. Because each tone lasts 300 milliseconds, that entrains an oscillation at 3.3 hertz. And however, these oscillations does not contribute more to the repetitions of an identical sequence. To unpack this a little bit, I know I'm packing in a lot of information, we can look at the phase coherence for the identical set of trials and similar set of trials. I think this is my last slide. And so what you can see is that for both of them, you have almost very high level of phase coherence, and that is similar between identical and similar set of trials at the frequency of tone presentation and its harmonics. Whereas for the low frequency arrhythmic activity, you have additional phase coherence for identical repetitions of a sequence above and beyond similar sequences. So what this shows is that the phase locking at the brain oscillation of 3.3 hertz and its harmonics reflect phase locking to the onset of each tone, regardless of sequence identity. However, sequence tracking is carried out by low-frequency arrhythmic brain activity. So I think I'll just end on this last slide. We've heard from Christoph today about scale-free dynamics in EEG microstates, and I've mostly focused on scale-free dynamics in low-frequency arrhythmic activity. And as I mentioned earlier, there is also we also know that amplitude fluctuation of brain oscillations have scale-free dynamics, and there is neuronal avalanches as well. So I think a very important question currently for the field is how are these scale-free dynamics at different levels connected? And there have been a couple papers on this too that I'll include in my slide that will be posted that people can read if of interest. And the negative sharp transients I mentioned is probably a link between these two. And I think it remains an open question, and much needs to be done to consider to provide an integrated framework across these different scale-free dynamics. And Matthias' group has done some very nice study about this link as well. So I think I'll stop there, and here is the take-home messages. Sorry, I went a couple minutes over.